talk 2 days ago, I mentioned already that the basis of our injury hypothesis, or the basis is a conclusion drawn from data obtained during the Munich SOD trial, published in 94. However, the real story goes even back to the late 60s. And during this time, I was working in Walter-Brendel's Institute of Experimental Surgery at the Munich LMU, I should say, LMU University in Munich. And in fact, during this time, the development of this model can be seen as a result of an outstanding interdisciplinary team spirit that prevailed at this institute during this time. For example, colleagues working here in the field of hemorrhagic shock, resuscitation regimens, shared their ideas, their knowledge, their study designs with colleagues from transplant immunology, and vice versa. And it happened that 20 years later, Konrad Messmer, you see here, it was the team leader of this team here, hemorrhagic shock. Now he was director of the Division of Experimental Surgery in Heidelberg, asked me, now the head of the Division of Transplant Surgery at the LMU University in Munich, he asked me to perform a clinical trial on kidney transplant patients using a free radical scavenger. And this, at that time, it was very popular, this superoxide dismutase, the SOD. And it was already known at that time that superoxide dismutase was a very active scavenger to remove the free radicals generated during reperfusion of organs and leading to the classic reperfusion injury. Well, during this time, I've written it down here, perhaps you can read. It was believed that the superoxide dismutase acts as a recovering substance during reperfusion of the body after successful resuscitation of patients in severe shock. And the interesting thing is this was the real marketing interest of the pharmaceutical company sponsoring this trial. In other words, the reperfusion of kidney transplants in the study patients should just serve as a model for this resuscitation scenario. And the idea behind was that improvement of early allograft function by SOD would imply that this radical scavenger, when added to recovery solutions, could improve organ function after successful resuscitation of patients in shock. So this was the real background why doing such a trial in kidney transplant patients. Well, with this in mind, then the trial was conducted, was successfully finalized, and finally published here in Transplantation, 94. And as expressed by the title, we found a beneficial effect of human recombinant superoxide dismutase on acute and chronic ejection events in kidney transplants recipients, as here expressed. So perhaps a few words to the study design. 200 milligram SOD was given once intravenously during surgery, a few minutes before allograft reperfusion, as you can see here, to patients who subsequently received cyclosporin-based immunosuppressive therapy. So very briefly, this kind of design, and acute and chronic ejection events were monitored up to a period of up to 8 years. Here you see the original data from the trial as published. Patients who received SOD, here shown in blue, showed a statistically significant reduction in incidence of acute rejection episodes down to an 18.5%, and irreversible graft loss down to 3.7%. And as you can see here, the long-term results were also significantly improved. And the beneficial effect was even demonstrable 8 years after just one syringe of superoxide dismutase. So, in other words, treatment of a non-specific allograft injury, here the reperfusion injury, resulted in a significant reduction in incidence of specific adaptive alloimmune events, here allograft rejection. And we concluded, in view of this convincing data, in terms of an argumentum e contrario, it is a tissue injury that initiates immunity. Well, at this point, I also have to mention the bitter pill of this trial. Namely, the original aim of the trial, the improvement of allograft function by SOD, was not significantly achieved. And from this point onwards, very disappointingly, the pharmaceutical industry lost any interest in further testing of SOD in the field of organ transplantation. I will come back to this problem at the end of my talk. But at first, let's take a closer look at the meaningfulness of our working hypothesis as published in '94. And as labeled here by this frame, we described an immune system, an autonomous, independently functioning immune system operating in humans, as you can see here, activated by sterile injury, here reperfusion injury, and after activation leading or eliciting an adaptive immune response, here an alloimmune response leading to allograft rejection. And in the center of this system, this immune system, we proposed a role for activated antigen-presenting cells, later appreciated to be dendritic cells, operating, so to speak, as a bridge between injury, sterile injury, and adaptive immunity. Also, but this is not the topic of today here, we discussed the possibility that certain products of the alloimmune response would contribute to the development of chronic rejection. So, in sum, from where we stand today, in '94, we described an immune system in its own right, preceding, activated by injury, preceding adaptive immunity, but unfortunately, probably we missed to call it innate immunity. So bad luck, now to speak. But then 2 years later, in this paper here, at least we called this system natural immunity, a little bit progress. Well, as you can imagine, during that time, I felt very, very lonely because nobody believed in our hypothesis. Many friends from immunology, they told me, they warned me, Walter, okay, you are a good surgeon, but please keep away from immunology, you know. Perhaps you can imagine when you are a surgeon, oh, I have such a thing. Yeah. And then Polly came along, look at this. And she saved me from falling into a deep depression. And she helped me to ignore the disregard for this model that had been raised by many immunologists, including German immunologists, but perhaps still are raising this. So then, sometimes later then, Ralph Steinman, you know, the founding father of dendritic cells, he was also of great help for me. You can see him here on a photo also taken at Surspoly, at one of those winter meetings. I think it was 1 year after you were there. And he was of great help for me. And actually, he told me everything about dendritic cells. He even proofread my manuscripts on dendritic cells before I submitted them. And perhaps you know this, very tragically, Ralph Steinman, he died just a few days before the Nobel Prize was awarded to him. So he did not, he could not get it anymore, that the Nobel Prize was given to him. fantastic man. Okay, now, thanks to this great support I experienced during that time, also received by many friends from immunology, I slowly was being prepared to refine our working hypothesis. For example, by publishing this paper here in 1999 and proposing that it is the post-ischemic reperfusion injury to allografts that leads via upregulation or via activation of dendritic cells to an alloimmune response. And this concept was even here illustrated in this figure here. And here, look at this in what I wrote at that time. The concept that PRI, post-ischemic reperfusion injury, activates dendritic cells and their function in alloimmune responsiveness fits nicely into Matzinger's hypothesis. 99. Then in 2002, 2003, in view of growing reports on the role of the innate immunity in infection, we transferred and applied this concept to injury-induced allograft rejection. And we coined the new term here, innate alloimmunity, published here in this paper here. And in this paper here, we described the term DAMS, standing for damage-associated molecular patterns. Also here in this paper, we accordingly, we wrote this sentence here. Damage-associated molecular patterns, such as heat shock proteins arising in the stressed allograft, serve as endogenous ligands for and interact with toll-like receptors on cells of the innate immune system, such as donor or recipient-derived dendritic cells and donor-derived vascular cells, and by this engagement activates them. Also in this paper, we designed this figure here, this illustration, and we sketched here a dynamic, sequential, cellular and molecular pathway showing that oxidative injury-induced DAMS here, via activation of dendritic cells, promotes an adaptive alloimmune response. And we stressed again that the DAMS-induced or DAMS-triggered upregulation of co-stimulatory molecules here, along with signal 1, is needed to fully activate a T cell, and then therefore initiate a T and B cell-driven alloimmune response. So again, this was the concept beginning 2002, 2003. Well, you see here, we exemplified DAMS by heat shock proteins. And the reason for this was that a year ago, we discovered a member of this family of heat shock proteins in reperfused donor organs. And this is an experiment that will be presented by Dr. Arbogast in a minute. But before closing, let me just come back to the problem I mentioned already. The reluctance of the pharmaceutical company to further test superoxide dismutase or another free radical scavenger in large-scale studies on transplant patients. Because, on the other hand, the data from the ASODATE trial showed or mirrored a dramatic therapeutic effect regarding the incidence of acute rejection episodes at 1 year in patients under cyclosporin-based immunosuppressive therapy. Just one syringe of 200 milligram SOD, of course no side effects, is equivalent to the administration of 2 grams of mycophenolate mofetil, that is accumulating dose of 730 grams mycophenolate mofetil per year. Just one syringe of SOD. And that would mean, of course, if we could apply such SOD, or if we could apply another free scavenger, edaravone, for example, is a very potent free radical scavenger, that would lead to a considerable reduction in incidence of immunosuppressive drugs as are being used today. And of course, fewer adverse effects. And not to mention the cost savings. So, well, I remember we, and including Polly, we approached several companies, key companies, and we confronted them with this problem, but there was no chance. It was frustrating. And we both had to learn one syringe of SOD, that's no business. So there was no chance. On the other hand, personally, I consider this at least an almost scandal in our ongoing efforts to search for new, better immunosuppressive drugs for our patients. But okay, let's move away from this dark side of this trial and move to something more enjoyable. Please, Helmut, it's now your turn now.
Thank you, Walter, for this fantastic introduction. I'm still very proud to have been, 2003, I have been with you together at Pflaum's Posthotel Pegnitz. This is the place where the term DAMPS was coined. So, historical meeting, I must say. I also had a glass of wine with Jules Hoffmann, who later became a Nobel Prize winner, together with Ralf Steinmann and Bruce Beutler. So it was really, and Polly was there, of course, and you two of you coined this expression. Then, yeah, at breakfast, that's correct. Then this was 1 year before, so the DAMPs were not known yet. The background you have already heard. I can go very fast over it. I will give you a short introduction and show you how we discovered the heat shock proteins in human organs, kidneys and pancreas, and show you the results and the conclusions. The working hypothesis was already nicely shown by Walter Land. Reactive oxygen species mediate injury to allografts activates the donor's and recipient's innate immune system that precede recipient's adaptive alloimmunity. And we postulated that the generation of reactive oxygen species during reperfusion injury causes the appearance of oxidized proteins in the donor organ cells and induce a heat shock response with an expression of heat shock proteins, and these should interact with the toll-like receptors of the cells of the innate immune system, such as dendritic cells and vascular cells. And this activation of the innate immunity was supposed to activate the adaptive immune system. And I don't have to go through this scheme again because it's almost the same as Walter already showed you. And there were two legs: the activation of the antigen-presenting cells on the one hand for the adaptive alloimmune response, and then the injury of smooth muscle cell proliferation that activates vascular cells and leads to alloatherosclerosis. So heat shock proteins have been shown. At that time we knew already that they serve as putative endogenous ligands for toll-like receptors. They are usually chaperoned outside the damaged cell and presented to the toll-like receptor bearing cells. And they have been shown to be introduced by oxidative stress. But nobody had shown before that during reperfusion injury, heat shock proteins are produced. So we looked for the reactive oxygen species-induced expression of heat shock proteins in the donor organ before and after cold storage, as well as before and after reperfusion in the recipient. This could be presented on this famous meeting in Miami, chaired by Camillo Ricordi in 2002, Everybody who was there knows that they wanted to go to Argentina first, but due of a money problem in Argentina, like they have had many times afterwards again, they had to change the location, and Camillo Ricordi, very shorthand, took over the Congress. So this was the what we did. First of all, we took blood from renal veins, and after 1.5 seconds, we put it into a radical scavenger called DEP-MPO, and another second later, we froze it in liquid nitrogen. It was a kind of a strenuous work during the operation procedure, but we had, in that case, we had saved our specimen for analysis for EPR spectroscopy. And these are data that were published by Jan Veertman and in our group. He was the EPR spectroscopic specialist, and he could show that after reperfusion, 5 minutes and 15 minutes after reperfusion, there was a peak of hydroxyl radicals to be detected in our renal venous probes. Now, could these reactive oxygen species activate heat shock proteins? And we found we took biopsies from 28 organs, a total of 25 kidneys and 3 pancreata in 24 patients, and had 36 renal biopsies, 20 reperfusion studies at different times, and we had 6 pancreas biopsies, 3 in situ and 3 ex situ. Donor and recipient age, mean cold ischemia time, mean warm ischemia time are listed below. What did we do with the staining? And it was a very, I would say, a very special staining method that was invented in our pathological department by Susanne Arbogast, by the same name as my wife, but she's a different one. Susanne is a pathologist and made her career with Roche later, but she took this staining method. The probes were, the biopsies were paraffin embedded, then deparaffinated with xylol and ethanol in decreasing concentrations, and then microwaved with a TRS, a target retrieving solution, and then incubated with a special antibody, HSP6B3, that was specific for the inducible heat shock component HSP73. And these heat shock component 73 could be detected in a dilution of 1 to 150 at room temperature with a double staining method called APAAP, which names alkaline phosphatase, anti-alkaline phosphatase, and was incubated with a linking antibody. This is shown on the right side with a brown symbol, and then was stained with fast red. Now I show you the results in the kidney biopsies. Post-mortally donated kidney biopsy after 23 hours of cold ischemia time is shown on the left side before reperfusion. Before reperfusion, we had a nice staining of the heat shock component HSP73. And on the right-hand side, you see the maximal expression that was achieved in another kidney, 16 hours cold ischemia time, 60 minutes after reperfusion. And if you look at the brain-dead donor, look at this biopsy on the left-hand side. Already in situ of a kidney biopsy in a brain-dead donor without ischemia or retrieval trauma, we showed a staining for the inducible heat shock component HSP73. And the negative control usually is done with skin biopsies, but we wanted a better control, so we took biopsies from healthy donors, like in a living donation. And here you can see that there is no heat shock response detectable. Even more expressed it is in the pancreas allografts. Here on the left-hand side, you see a pancreas biopsy in a brain-dead donor. Already there, the Langerhans islets are staining very nicely. Less staining in the exocrine apparatus, mainly because there might be digestion of the heat shock component. But on the right-hand side, you see an ex situ biopsy of a pancreas that was not transplanted after retrieval, and we left it 6 hours in the warm. So here we had a very nice staining of the heat shock component in the Langerhans islets. We concluded then that the inducible heat shock component HSP73 is expressed in postmortally donated renal and pancreatic allografts already after organ removal from the donor, after cold storage, and even more after post-ischemic reperfusion of the graft in the recipient. HSP73 expression steadily increases from the time of removal in the donor up to the implantation in the recipient and culminates 60 minutes after start of reperfusion. And this is a stress protein that is already expressed in the brain-dead donor, and what also can be said, it is upregulated during cold storage, particularly after post-ischemic reperfusion of the graft. So this initial hypothesis has been proven, and I think that we have been the first describers of an inducible HSP73 in post-ischemic reperfusion of allografts. And here I thank you for your attention. Yeah. Sorry? Yeah, a few questions, of course. Any questions from the audience?
Thank you. So I wonder, is the heat shock protein inside or outside the cell? Because it needs to be outside to be recognized by the immune system, I guess.
It generates in the inside of the cell and is chaperoned outside. Okay. And presented to the antigen-presented cells through the membrane. So the toll-like receptors from the dendritic cells dock on this presentation of the heat shock component. This is a mechanism that has been known. This is nothing that we have proven, but we have shown that it exists in a brain-dead donor, that it is upregulated after reperfusion and during cold storage.
And maybe another question, if I may. The effect of a one-time SOD injection prevails for years. Did I understand that correctly? Like more than 8 years, correct? So how is that possible? There has to be like some type of cellular memory, or is there a mechanism, a cellular mechanism that's kicked off and prevented by SOD?
Can you formulate very brief again this question?
Oh, the question is that how is it possible that this one-time SOD injection prevents a mechanism of immune response over years that is still visible, like more than 8 years after the transplant?
I have an idea. Perhaps Polly doesn't agree, but my idea would be that we have perhaps induced T-Rex. Because what other mechanism would operate 8 years after one injection? Yet, no. You will get the answer.
So, I happen to think differently about Tregs from most people. Maybe we'll get to that. Another possibility is that in the absence of damage, lymphocytes get, for simplicity, signal 1 without co-stimulation and die. So you do a huge reduction of the number of cells that can recognize the graft. And if you do such a huge reduction, then of course rejection doesn't happen. Now that would also depend on the age of the recipient, because the thymus can make new T cells in young ones. But then again, if the graft is quiet, that should induce tolerance as well. I don't think you need to bring in anything like Tregs.
Okay.
Can I say something just for fun? Just for fun. There's a woman in Denver whose name I can't remember at the moment, who acutely stressed rats and then injected them with bacteria, IV. And she found that, and then took the blood and did cultures for the number of bacteria that existed, 6 hours later. And she found that acutely stressed rats had much lower numbers of bacteria. And she asked what molecules were important in this. And she looked at all kinds. One of the stresses was simply to put cat food on top of the rat boxes and allow a cat to walk there, for those of you who are interested in the psychology of transplants. Right? Just the cat stress was enough. And she found that it was inducible HSP70 that was alerting the immune system to clear, innate immune system to clear the bacteria.
Highly interesting. Thank you.
Yeah, very interesting.
I remember her.
Yeah. Okay, Helmut, can we proceed? Or
I think we should proceed now in the interest of time. And now it's time for one of our two special guests.
You think about it, you try to work with it, but so many other immunologists have not. People tell me, I teach the danger model. I say to them, great, what do you teach? And they say, this is an immunology course. They say, in the chapter on initiation of an immune response, we talk about dendritic cells, and by the way, macrophages also, and how you need to activate them. And if you don't activate them, you don't get an immune response. And you can activate them with PRRs, PAMPs, and also DAMPs. So injury-induced, damage-induced molecular patterns. I say, great. What do you teach about why a woman doesn't reject her fetus, the other kind of transplant? And they say, oh, we all know that pregnancy is immunosuppressive. And I say, you are not teaching the danger model. How many of you think that pregnancy is immunosuppressive? You're taught that. Peter Medawar, who won the Nobel Prize for doing the experiment that put the self-non-self model on the map, understood that pregnancy was a real problem because fetuses are foreign and you ought to reject them. And so he came up with a couple of ideas, and those are still in the textbooks. The first one is that pregnancy is immunosuppressive. This is really smart. You take the individuals carrying the next generation and you turn them into AIDS patients. Right? And when something is evolutionarily dumb, I have found in my career that it's usually wrong. So how many of you have carried fetuses? Yeah. Did you have to live in a bubble? Were you sick a lot? No. Did anybody notice? The second thing that Medawar suggested, also in the textbooks, is that the placenta is a physical and immunological barrier between mother and fetus. Also not very smart, because if your fetus gets infected, there is nothing you can do about it, and it's dead, and so are you. Also not true, right? We know that fetal cells traffic into the maternal circulation. People isolate those cells in order to do non-invasive prenatal diagnosis. And you guys, at least the Dutch, know that maternal cells go into the fetus. So some of you know that at Eurotransplant, they discovered that people waiting for a kidney that had a living donor first are always tested to see if they already have a strong response to the donor, because if they do, you don't do that transplant because it's dead in 2 weeks. But they discovered that about half of their people waiting for a kidney made no immune response to the non-inherited transplantation antigens of their mothers. They reacted to their fathers, but not their mothers. That was Jan van Root. And Lee Zhang and Rick Miller followed this up in rats, where you can mark the father's cells, the mother's cells, and the babies separately. And they found that 50% of adult rats carry micro amounts of cells from their mothers. 50% of SCID babies, the boys who have to live in a bubble, have T cells from their mothers, small numbers. So there's traffic from fetus to mother, and there's traffic from mother to fetus, but the fetus is not rejected. So why? So the danger model says it's because it's normal, the cell death that goes on during development, you kill the cells to make fingers, for example, is normal programmed cell death. The cells don't release alarm signals. They're picked up by scavengers. So as long as your fetus is developing normally, there is no activation of an immune response. And should that fetus get infected, you do react. And in fact, a common knowledge in medical circles is that infection during pregnancy can result in spontaneous miscarriage. That's the immune system working. So that's a transplant, which is normal, doesn't release DAMPs, and you don't reject it. Right? And yet people still teach that pregnancy is immunosuppressive. So let's take another transplant, tumor. It's not really a transplant, but it also has foreign antigens. It has mutated antigens. It has embryonic antigens sometimes that are upregulated that normal tumors don't have. Why don't we reject those? So what's wrong with a tumor? Anybody? What's wrong with a tumor? What is the primary problem with a cell that becomes a tumor? It has lost growth control. In the beginning, there's not much else wrong with it. It's just multiplying or dividing, since those are the same in biology. And is this cell sending alarm signals in the beginning? No. So what happens to a T cell that can see a tumor antigen in HLA? No alarm signals, no activated APCs. This T cell has a receptor that can see that. It's going to get signal 1 without co-stimulation and die. So in the beginning stages of a tumor, you don't have an immune response. Now, if this tumor gets really big, and it's a solid tumor, and it's poorly vascularized, you will start to get some death in the middle. But starvation death is also programmed cell death. And so there's still no alarm signals. But if this death eventually gets to be so bad that it overloads the local scavenging capacity, now you will get some DAMPs. But what has happened to all the T cells that can see that tumor in the meantime? They've been deleted. You have 3 left. They valiantly try. They get activated, they multiply, they go out and kill. Each killer we know can kill between 30 and 100 targets before it needs to be reloaded. So the killer that gets activated by this overwhelming death goes out and kills 3 to 30 tumor cells. And then it has to go back into a draining lymph node and see an activated APC presenting a tumor antigen. And then it has to spend a couple days multiplying, and then it goes back out. What is the tumor doing in the meantime? It's making millions more cells. You have lost the race. Yeah? So people know this in tumor immunology. They have tried many things. They immunize or use blocking antibodies, or they isolate cells from the tumor and grow them up in bathtubs full of IL-2 and then put them back in. And all of those treatments work for about 30% of tumor patients. And so people keep trying to look for more active, better tumor treatments. And what I'm trying to tell them, and unlike you, they're not listening, is that it's not that the treatments are bad. It's that they're using them incorrectly. And they're using them incorrectly because they're still using them based on the old self, non-self model. Here's how that works. In the old model, if there is something foreign and you can activate the immune system to it, the presence of that molecule is enough to keep the immune response going. But we now know by our models that this is not true. What happens with an immune response? All right, let's say, oh God, quick. This is very transparent. Let's say you have a tumor growing and you do something. You vaccinate, you give an adjuvant, you add tumor-infiltrating lymphocytes, whatever, and it's effective, so the tumor gets smaller. And if you're lucky and you get the last cell, you're good. But if you're not lucky, what's going to happen? The immune response that you elicited goes up and it comes down. Killer cells peak at day 8. They're gone by day 14. They're not gone, they're just resting. They need to be reactivated. So your tumor starts to grow. So now you do it again. And you get another quicker, higher immune response. And you get to, And if you're lucky, you got the last cell, you're good. But if you didn't get the last cell, that tumor starts to grow. And what happens is the physicians stop. Why do they stop? Because they're thinking in old vaccine mode. You make a vaccine, you activate the cells, they expand, you boost, you activate them again, they expand some more, that's about it. There are mechanisms that prevent forever expansion. You know, your lymph nodes can't get this big. Good timing. When you do that for a virus infection, it's great. Because when you boost, the immune response goes up, it comes down, the cells go into resting mode, and when the pathogen comes in, it does the damage that reinitiates that response. But killing by killer cells does not. It induces apoptosis in the targets. The immune system does not throw fuel on the fire. It has to know when to stop. So when you give a vaccine for a tumor, you need to keep going. Because the tumor death by killers is not going to boost the response. It's programmed cell death. The cells get scavenged. They don't release stamps. So you need to keep going. You need to keep going. And you need to keep going until the last cell is gone. And because the tumor immunologists have not learned this somehow, they don't do it. About 8 years ago, Steve Rosenberg used to give these talks about TILs, tumor-infiltrating lymphocytes. There are some people who have melanomas who have cells in them trying to kill the melanoma, but there are not enough cells. And so what Steve did is he took the cells out of the melanoma, he grew them up in the lab, and he put them back in. And he had this beautiful set of slides. A patient with a melanoma metastasis in the lung that was the size of a softball. A softball is like a baseball, but it's bigger and softer, and they allow women to play with it. And he took the TILs, he put them back in, and the tumor went down to the size of a normal baseball. He did it again, and the tumor went down to the size of a golf ball. And then he stopped. And the tumor grew out and killed the patient. And I asked Steve, why do you do this twice? Why do you not keep going? And his answer was interesting. He said, I have so much money, I have so many clinical fellows, and I only have so many tumor-bearing patients that are willing to give their bodies to science, I want something that's going to work in 2 shots. And I say to him, ain't going to happen. You could get lucky. But most of the time, you're going to get unlucky. And what we see today is that these people get lucky with about 30% of their patients. And they wonder why the other 70% are not responding. It's because they're still stuck with the old model. What you think really matters to what you do. So that's the other transplant. And now I'm going to give you a little something that has nothing to do with transplantation, but it's an aspect of the danger model that I've been thinking about for a little while. And that is an old problem. What makes an allergen an allergen? Why is it that the cat, which makes hundreds of thousands of proteins, one is a main allergen, and there's another 3 that are kind of minor allergens. An allergen is a molecule to which you respond by allergy. Yeah? What about all those other ones? What makes those special? What makes an allergen an allergen? The house dust mite makes thousands of proteins. 3 are allergens. The birch tree makes thousands of proteins. 2 are main allergens. What makes an allergen an allergen? Right? So I think the danger model gives us an idea. There are 4 categories in my mind of allergens. 1, the molecule causes damage. The main allergen of house dust mite, DIRP1, is a protease that attacks the surface of lung epithelium. It's not surprising that you make an immune response to it. There are many others. 2, it's packaged with something that causes damage. It's something that causes damage. Bee venom. Phospholipase A2 is a phospholipase. It destroys membranes. There's a melatonin in bee venom, which it's a hydrophobic peptide that makes holes in membranes. It can destroy red cells. So you're packaged with something that causes damage. 3, it mimics an endogenous or exogenous damp. So in bee venom, there is also a molecule that mimics MD2. And MD2 is a co-molecule for TLR4. It binds LPS. So if you get a lot of MD2, you now start to respond to just the normal amount of LPS that leaks out of your gut from the bacteria in your gut. So you mimic either an endogenous damp or an exogenous damp. And 4, you are given, or you get it, at a dangerous time. Now, what do I mean by that? My favorite one here is peanut. All right, so here we have DIRP1. Here we have bee venom. I can't hold the microphone and the past transparency, and okay. Here we have MD2. So what about peanut? I don't know if you have peanut allergy in Germany, but it's a huge problem in the United States. And it didn't happen until the 1990s. And if you go to Indonesia, kids are eating peanuts all the time. And I grew up in Holland, where we eat peanut butter for breakfast. Yeah, on sandwiches, on bread, good bread. So what happened in the 1990s that we started getting peanut allergy? Sometimes lethal peanut allergy. Well, it turns out the Canadians learned this first. They have banned peanut oil in baby creams because they saw that many of the children who become peanut allergic had eczema as babies. So they started looking, and they found that peanut oil is a wonderful oil. It's in many creams. So now you have a baby that has a rash, damaged skin, activated APCs, and you slather it with peanut. Is it any surprise that they make an immune response? So you're given at a time of danger. The anecdote which proved this for me is a couple at the NIH, Dan and Kat Barber, who had a baby who had eczema, a lot of rash, and they knew about this. So they went to, health food stores, and they got baby creams that used sesame oil instead of peanut. And when their baby was 1.5 years old, in his daycare, he anaphylaxed when they fed him hummus, which has sesame in it. So if you have a baby with bad skin, give them mineral oil. It works, and it's much easier to avoid than peanut. So I'm out of time. The men tend to kind of go over. But I'll stop now and take questions. Any questions about anything about the danger model you want to ask?
Polly, thank you so much. Not only the presentation method was original and I would say kind of unusual, but also the topics that you chose. But everybody understood what you were talking about. There must be questions from the audience. Please.
So if you have a pregnant woman and you do surgery on the baby, I don't know the English word, you do surgery while she's pregnant on the baby. You can do surgery inside. You would do damage to the baby. So according to your theory, this would lead to an immune reaction.
I bet you get one. In fact, you could test that.
But there is surgery in unborn babies.
Yes.
So
I bet you get a transient immune response. Right? You're going to get a transient immune response. Up, down. When that baby heals, remember that killing by a killer does not induce DAMPs. That was shown by Bill Heath in Australia. So you're going to get a transient immune response. Look for it. I'll give you another example. There's a funny thing in lupus. I didn't talk about autoimmunity, but I wrote an article on autoimmunity. It's in Frontiers in Immunology. It came out last year. Women who have lupus, when they get pregnant, not all, but many, stop having lupus flares. So that's an interesting thing about pregnancy. Now, it also turns out that there are women who don't have lupus, who get lupus while they're pregnant, and then it stops when usually they miscarry. So how can the danger model explain both of those? So here's how it does. Right? 1, there's a lot of death that goes on in female bodies that doesn't go on in male bodies. We pop an ovary every month. We kill off, clean up, and regenerate an entire uterine lining every month. That's a lot of death. That's a lot of scavenging. It's a lot of genetic control necessary. If a mutation happens in that process, it could easily lead to release of DAMPs. So I tell rheumatologists, if any of you have friends who are rheumatologists, tell them if they get a young woman that is recently diagnosed with lupus, put her on the pill or give her one of those long-term injections and don't let her cycle and see what happens. Now, I've been telling rheumatologists this for years, and they say it's not normal. I say, you know what? It's not normal to cycle every month. Until the pill, we were either pregnant or lactating most of the time. And women in third world countries who are not on birth control are pregnant or lactating most of the time. And they menstruate about 10 times less in their lifetime than Western women do. It is not normal. So put them on the pill and don't let them cycle and see what happens. It's called diagnosis by treatment. So what happens in those women who don't have lupus, who get lupus when they're pregnant? And this comes back to damage to the fetus. What I think is happening, and it's hand waving. People sometimes ask me if my wrists get tired of hand waving. There's something wrong with that baby. There's a lot of death during fetal development, and that's a lot of genetic control necessary. And you could also have environmental toxins that get in the way. So I'm going to predict that that baby has something wrong with its death processes. And so it's releasing DAMPs. And those are getting picked up by the mother. And so she is getting lupus because of bad death somewhere in the fetus. These fetuses are usually miscarried, which tells you something. So you get this transient immune response, lupus, in the mother, and it goes away when the baby goes away. Do the experiment. I bet you, when you do surgery on the unborn, you will get a transient immune response. It's a prediction. Mel Cohn, who I think was one of the greatest immunological theoreticians, he invented signal 1 and 2 out of the air, says, it doesn't matter if you're right or wrong. Go out and be clear. Make predictions. If somebody wants to prove you wrong, they'll do the experiment. And in the process, we'll learn something we wouldn't have learned otherwise. Do the experiment.
More questions from the audience?
Is anybody thinking about RH? RH negative mothers make responses against RH positive babies? No? Okay, I won't talk about it.
Here's another question.
Important question would be how can we make use of this knowledge in terms of transplantation? How do we flush out the dams from the kidney we are about to transplant?
Well, I think there's so many, DNA, RNA, HSPs. There are so many dams, you're not going to be able to block them. But they go through a bottleneck. Right? What they do is they activate antigen-presenting cells. And we talk about dendritic cells, but no, people forget macrophages. They also do it. So you block co-stimulation. Does anybody know Alan Kirk? Captain Kirk, actually Commander Kirk, believe it or not, he was in the Navy. He was a transplanter, and he did some really interesting studies with monkeys, kidney transplants. He took monkeys, gave them a kidney, after testing them to see that they had strong responses to the donor. So he's working against himself. Did the kidney transplant. So it's a living donor kidney transplant, which is always better than the dead ones. And he blocked signal 2. So cyclosporin blocks signal 1, and that's important. Again, what you think influences what you do. So, thank you. So you have your antigen-presenting cell presenting HLA plus a peptide, right? T cell. The T cell gets signal 1 from binding, and then it gets signal 2 from the activated APC. Nash. I should put some tape here. Okay. So this is simple. This is simplified. Signal 1 is signal 1. We know that. Signal 2 is actually a blend of co-stimulatory and co-inhibitory molecules. T cells are really much more like NK cells than we think. They get positive signals and they also get negative signals. The positive signal 2s are what activate them. And so what Kirk did was he blocked CD40 ligand, CD154, which is a receptor for signal 2. And he gave this to the monkeys for 5 months. At first daily, and then every week, and then, I can't remember exactly, every 3. And then at 5 months he stopped. And they didn't reject. This is a Nature Medicine paper. I was a referee, which is why I know about it. They didn't reject. You block signal 2, and you mimic the body's normal way of inducing tolerance. Right? When a T cell sees a healthy tissue, not an activated APC, but an unactivated APC or a healthy tissue, it gets signal 1, but it doesn't get co-stimulation, and so it dies. And so if you want to apply this, do what Alan Kirk did. Now, what happened with that, there's always something that happens in medicine, right? It turns out that people who have atherosclerosis were being given this anti-CD154. And because CD40 ligand is also on platelets, they were getting clots. And so the trial on the kidneys was stopped. And it's too bad, because my brother died of hepatitis C, kidney failure. I tried to give him my kidney. I was going to go with this clinical trial, and it was stopped. And then they discovered that I'm a complete mismatch. So they wouldn't do it. That's what I would do. Block the DAMPs as much as you can. Do as little damage as you can. Do the SOD. Oh my God, do the SOD. This is a political problem, not a medical problem. And I don't understand. Well, I do. One shot of SOD costs pennies. The pharmaceutical companies are not going to make any money doing this. What? That is the reason. But here's the thing. I tried to talk to the NIH about this. When there is a potential medical treatment which is cheap, too cheap to get the pharmaceutical companies interested, the government should support it in a country that has government-supported medicine. The insurance companies should support it in a country like the States, right? Because if they don't have to pay for the cyclosporin, the Mofetil, everything else you use, they're going to make money. At least they're losing less money. Right? We need to get that message across. Again, what you think influences what you do. So we need to get across the message that when you've got something simple and cheap, forget the pharmaceutical companies. That's a bad model for these things. Use the government, use the insurance companies.
Well, there are many cells each day with have mutations and are killed by the immune system.
Really?
Yeah, I think so.
What evidence do you have for that?
Just lectures.
Yeah, well, what evidence do they have for that? So it used to be thought that we got a tumor every few minutes because cells are dividing and they're mutating and da-da-da, and that the immune system killed them, and that when a tumor grew out, the immune system had failed somehow. But then somebody found a mouse with no thymus. The nude mouse also has no hair. And it gets no more tumors than normal mice. And then the rag knockout mice was created. No T cells, no B cells. So the nude has no T cells, but it still has B cells. The rag knockout has no T cells and no B cells, and it does not get more tumors than normal mice. So this idea that we are constantly making mutated cells and the immune system gets rid of them does not fit with those data. So when somebody tells you that story, ask them, what is their data?
Time for one more question. Otherwise, I would.
I'm going to tell you about RH, even if you're not interested.
Polly, thank you so much.
No. I'm going to tell him about RH. Then I'll down. You guys do. Usually somebody asks me about this. RH negative mothers that have an RH positive fetus, blue baby, real problem, mothers reacting to the fetus. So how can I explain that? The fetus is fine. It just has this RH antigen that's foreign. And I point out that God is in the details. The important detail here is that it's a problem in the second pregnancy, not the first. Right? Physicians know this. They give these women Rogam. It's an anti-RH antibody to block it during birth. Birth is dangerous. You have ripping and bleeding. Surgeons with knives, right? Dangerous time. That is when women get primed. Not during the pregnancy. They get primed at birth. So let's do a little bit less damage at birth, if we can.
Thanks a lot, Polly.
Yes, a topic of evolution. And please allow me, before we start and come to the second highlight then, to make a few introductory words on this topic. Well, some of you may still have a little doubt about the correctness of the danger-injury model. And I think this is okay, but perhaps Theodosius Dobzhansky, he may help you overcome your doubts. Theodosius Dobzhansky, an Ukrainian-American geneticist and evolutionary biologist, he stated, "Nothing in biology makes sense except in the light of evolution." So let's see whether the danger-injury model makes really sense when seen through the glasses of evolution. Well, I told you 2 days ago in my talk that DAMs not only play a role in medicine, in human diseases, they have a completely different function. And actually the primary function of DAMs, namely the current notions in evolutionary research hold that all creatures, all organisms on our planet rely on DAMs for their daily struggle for life. To use again Darwin's fantastic term here. From plants to invertebrates, from invertebrates to vertebrates. And together with Dr. Seong, Polly and I recently wrote an editorial on this topic. So let me use the following minutes to give you a few examples from this exciting area. For example, studies on Drosophila melanogaster showed that the cytoskeletal protein actin here is an evolutional conserved stem that signals tissue injury in the fly. So there, the Drosophila is working with stems. In studies on shrimps, another dam, the PRX4, the antioxidative enzyme peroxyredoxin 4, was shown to enhance bacterial resistance via the toll signaling pathway. So this dam, for example, is released by heat stress induced necrotic hemocytes of this animal. So another interesting study. Also in studies on corals, Amphioxus and flounder, the perception of another DAMP, extracellular ATP, was found to be highly conserved throughout multicellular animal evolution. And as sensing receptors, the researchers here identified certain so-called purinoceptors. For example, these P2X7-like receptors in coral and Amphioxus, and the P2X7 receptor in flounder. Perhaps you know this Amphioxus, it's a fish-like animal, in Deutsch Lanzettfischchen, glaube ich, genannt. Lanzettfischchen. And flounder is flunder, soll, glaube ich, sehr gut schmecken, wenn gebraten. Also alle diese Kreaturen verteidigen sich jeden Tag mit der Generation von Dams. And let me close with another striking example from evolution, the fight of light response of C. elegans. And per definition, this response is an active defense immune response in terms of a stress response. And the term stands, for example, for the two choices our ancestors had when confronted with danger in their environment, to either fight or flee. So let me just share with you a few experiments which, in my view, or in my opinion, impressively demonstrate the intrinsic nature of this flight response. And I am talking about the C. elegans worm. As you know, the worms, they live in the soil, whereby bacteria are their main food. So that means they have to distinguish between nutritious, harmless bacteria, their food, here shown in green, and dangerous, harmful bacteria, here shown in red. And really, the worms can really differentiate, can really distinguish between them, as evidenced by their learned behavior to avoid the harmful bacteria. And this reaction, this response, is called the pathogen avoidance reaction or pathogen aversion reaction in terms of a protective immune response. And more excitingly, this typical behavior of the worm can be reproduced in vitro experimental settings. Here you see this setup. You know, if you place the worms in a petri dish on a layer of harmless, non-pathogenic bacteria, you see they feel really comfortable, they tolerate them, and they feed on them, so they stay there. However, if you place the worms on a culture medium containing harmful pathogenic bacteria, they, after a while, look what they are doing. They flee, they disappear. They avoid them. So the question, of course, is, or the researchers, they interpret this as a stress or injury-triggered pathogen avoidance or pathogen aversion in terms of defensive flight response. Well, in view of this typical behavior, of course, the question is, how can the worm recognize the pathogenic bacteria? And the answer of this question is provided by recent studies on Enterococcus faecalis, as you can see here. And the researchers found that it's the intestinal distension caused by bacteria that is perceived by the worm. So you can see here, here you see the unhealthy distended gut of the worm, here, compared here to controls. And interestingly, one could imagine that there is a debate now going on, a debate on the nature of the receptors that sense the pressure caused by intestinal distension. That means receptors which should be located here at this place, at this location here. And then send signals to the nervous system to elicit such a pathogen avoidance. So there's a debate going on. And for example, the researchers here in these two papers, I just selected them for you here, they discuss a possibility that it is the intestinal attention that generates certain as yet unidentified stems. So at least they are already discussing that stems may play a role, as you can see here. And these stems then are sensed by a highly conserved receptor, so-called FSHR1 receptor. This is a receptor from the G-coupled protein receptors. And then after recognition, this receptor triggers a pathway to the nervous system, which finally results in aversion. So this, as you know, some discussion is going on. It is not exactly known. Well, but I mean, regardless of this poorly defined signal mechanisms, the key message of these experiments is as follows. If the bacteria are harmless, as you can see here, they stay there, they are well tolerated, the worm is very comfortable with them. Only, or I should say, no, I should come back, please go back. The key message again, the pathogenic avoidance response of worms is not triggered by non-self bacteria per se. This is the key message. If they are harmless, as you see here, they are tolerated. Only if they are harmful, dangerous, then they are avoided. So, the
curiosity, how long do the worms remember?
I don't know, but they have to learn it. That is it. They have to learn it. It is not by birth already there. They have to learn that they are harmful. Yeah. But the question, of course, is what do these evolution-related experiments, what have they to do with the reality of everyday life? And here you will get the analogy. The commensals of our microbiome reflecting this harmless bacteria here in the Petri dish, they are 100% non-self, but they are not immunologically attacked. So what I am doing here is just to repeat what Polly has been trying to hammer in our brain for 30 years now. Because you see here the semi-allogenic fetus, 50% non-self, but as Polly told us, not rejected by the pregnant women because it doesn't send any danger signals. On the other hand, the pathogenic coli bacteria, for example, are damaging and they are immunologically attacked as diagnosed by colitis clinically, and they reflect these harmful, dangerous bacteria in the Petri dish. And here's the last example. Damaged allogenic non-self is rejected in the form of damaged transplants. And as I told you 2 days ago, every transplant inherently is a damaged organ. So a lot of but some now experiments related to evolution, they really give us some idea that probably the danger-injury model is really correct. And I think perhaps Theodosius Dobzhansky, probably he would agreeably comment in the following way. Polly, the danger-injury model makes sense in the light of evolution. So thank you very much. And before we now proceed to the second highlight, I will give to Helmut again for the introduction of Professor di Lorenzo.
Thank you. And it's really an honor to be here, to have been invited to this meeting and to meet Walter and Polly again. We met in Mexico a few years ago. And maybe we learned something from plant. That is the question mark. So let me just outline the main characteristic of the plant immune system. Are just the characteristics that are shared by protozoa and metazoa, and multicellular organisms that lack an adaptive immune system. So it's just an innate immune system. That means that it relies on germline, germline encoded recognition receptors. In plants, there are no circulating cells, no circulating antibodies, and plants are characterized by cells, each of which is able to mount an immune response upon recognition of danger signals. And the recognition apparatus is just similar to that we observe in protozoa and metazoa. So we have pattern recognition receptors. I still have to find the black. Pattern recognition receptors, and in plants, so far they are mainly plasma membrane receptors, and these recognize these widespread danger signals that are the MAMPs and also the DAMPs that are released upon tissue injury. These signals activate the so-called pattern-triggered immunity, and I will tell a few things more later. And then we have another branch of the immune system. Here we have a sort of very specific interaction, genotype specific. Effectors, genotype specific, are recognized by node-like receptors. Now the terminology is just the same as in the animal immunity. So these not membrane-bound receptors or intracellular receptors, these are the pattern recognition receptor. In plants, they are involved in triggering the PTI, the pattern-triggered immunity. Intracellularly, we have the NLR that in plants are involved in triggering ETI, ETI-triggered immunity, effector-triggered immunity. You see in this slide that there is one main message, that many elements, most of the elements, in plants, not all of them, that work in immunity have a special structural domain, which is the Lucy-Rich repeat domain. And you find it here either in PRRs or in LRs in a different position. So this is a main structural motif that mediate recognition in immunity in all these organisms. And you also see that in the intracellular receptor, the NLR, there is a striking similarity also in this case. If we look carefully, we can also see that we have also similar domains. For example, the TIR domain that is typical of the cytoplasmic part of the TLR, that in plants is used in a different way. You see, in this case, we find it usually at the N-terminus of the NLR. So there is a sort of commonality in the use of structural domain, but also a different way of assembling them to obtain an efficient defense response. And here, just a structural view of this Lucy-Rich repeat domain in plants and in animals. These are the TLR, and also the Lucy-Rich repeat domain in the NLR in plant and in animals. Something that I like to point out is that unlike vertebrates, which have a very limited number of toll-like receptors, if you go to plants, you see, and you just look at a very tiny plants, Arabidopsis, which is the model plants for the plant biologists, you can see that, that you have 600 protein with predicted topologies of receptor kinases. In rice, they are 1,000. And they are not only Lucy-Rich repeat, LRR, but they are also different kind. So there is an amplification of the recognition apparatus in plants. And let's say the conservation between plants and animals goes also downstream of the recognition events. Cascades, mitogen-activated protein cascades are activated in plants and animals. But again, if we look at the elements, there is a multiplicity in the system in plants also for the MAPK signaling cascade. Let's say 23 single kinases, 10 double kinases, 80 triple kinases, 10 kinase. This gives a tremendous potential for modulating the defense response in plants. All this redundancy has been associated with the inability of the plant to fly away, to escape. So they are sessile and they have to stay. So they have to rely to a very large apparatus of recognition protein. And just to give you an idea what happens upon the recognition, of course I go with the essential information. You have a local response at the site of infection, and you have, when the recognition occurs, sorry, I cannot catch this one. When the recognition occurs, you have really a very large battery of defenses. Here they are. The pattern trigger immunity. I don't go into a lot of details. There are proteins, antimicrobial compounds that are synthesized. There is a big burst of ROS and nitric oxide, which play a key role. And there is fortification of the mechanical barriers. And when you have the effect of triggered immunity, you have an additional response, which is a programmed cell death of a few cells just at contact of the pathogen. Is a sort of it has been interpreted as a sacrifice of a few cells that allow a more efficient defense response. A very recent paper actually says that in these dead cells, before dying, there is no activation of these responses, but the death of these cells is important to activate the defense responses around, and dams are being called in this effect. And this is a very recent paper. So beside the local response, we have also a systemic response. When you have immunity at the local level, you have an amplification and a signaling that is transmitted to distal tissues, and you have protection of the plant that can last even weeks or months. And this protection is against many pathogens. It is a wide spectrum resistance. This is called systemic protection, and this is an important response of the plant. But let's go now to, beside the introduction, to the focus of my talk here. The focus is our plant dumps, because plants have learned to respond to tissue injury. And here there is some history of the concept in plants. The observation that hydrophilic molecules in damaged plant tissue extract activate immune responses dates back to 1974. The group was studying wounding, the wound response. And later, a few years later, the active molecules, some of the active molecules were identified as oligosaccharides coming from the plant cell wall, galacturonic acid-rich oligosaccharides. This is 81. And the identification was in 1983, and these molecules were the oligogalacturonides. These are pectic fragments that derive from the pectin of the cell wall. This discovery led to the term coined by Peter Albersheimer's group in 1992 of oligosaccharin that indicates oligosaccharide with a biological activity. In animals, we heard already, we have the tissue, the notion that tissue injuries and danger induce immunity, up to the term damage-associated molecular patterns in 2003, that actually in plants replaced the term endogenous elicitor. Quickly, there are now many DAMs identified in plants, and some are shared between plants and animals, extracellular ATP, the HMBGB1 group of protein, and extracellular fragmented self-DNA. But as I told you, the oldest and better known DAMP are the oligogalacturonides. And these have been for years the focus of our work. So although not identical, not really shared, the there is a very nice parallel between the signaling system mediated by the extracellular matrix in animal and plants. There are, in both type of organisms, there are polysaccharides, acidic polysaccharides in the cell wall, in the extracellular matrix. In the plant, we say cell wall because it's thick, much thicker than in animals. And fragments of these acidic polysaccharides, one is hyaluronan, the other one is the homogalacturan, part of the pectin. And the fragments derived from these acidic polysaccharides induce defense responses. They also share an interesting feature that their action depends on their size. Only intermediate size molecules are active in immunity. This is particularly true in plants. Short oligogalacturonides, short fragments of homogalacturan are really at the moment are no immune activating activity or very little. Actually, there is a recent paper that they say where they say that they are suppressors, but it's a very recent notion. So, and the plant cell wall, the plant extracellular matrix, is a rich source of dumps. Besides oligogalacturonides, recently cellulose fragments have been found to act as dumps, and also hemicellulose fragments, mixed-linked glucans. Just to make a very long story short, because my group have worked for years on oligogalacturonides, they induce defense responses. If you spray them, they protect against pathogen attack. They induce very strong bursts of ROS. So they are potent activator of ROS in plants and activate transcription and reprogramming. So they have a lot of effect. So besides defense, we in our group also found out that oligogalacturonides actually usually a lower concentration than those that are active in immunity also antagonize an hormone involved in development. So they have the potential to regulate developmental processes. And in 1989, after some biochemical work, we found that the formation of oligogalacturonides actually can be favored by the interaction of a plant protein, the PGIP, polygalacturonase-inhibiting protein, and with the enzyme that produces the fragments, the polygalacturonase. So the interaction between the polygalacturonase and this inhibiting protein just leads to a stabilization during the time of the formation of the oligogalacturonide. This appeared to us as a very unique system. The plant counteract the action of a polygalacturonase to favor the production of oligogalacturonides, which are dumps. And here is just how we imagine the system might work. We have the degradation of the homogalacturonide by the polygalacturonases produced by microbes. So usually you go to short products that are nutrients for the microbe. But in the presence of the inhibitor, you have a slowdown of the interaction. This is what we thought. And you were favoring the production of these oligogalacturonides, which are active as dumps. We worked a lot on this. We did structural work, biochemical work, transcriptomic work, and we clarify how the oligogalacturonides really activate a very powerful defense response. I didn't include the production of ROS in this picture, but you have also a strong production of ROS. And now there is a really sort of a breakthrough just a few weeks ago, a structural work by a Chinese group that collaborated with my group uncovers that the release of oligogalacturonides by polygalacturonases and PGIP when they interact is not just a casual event, but is the result of a unique biological design. This paper was published in Science just a few weeks ago, 2 months ago. And just quickly, what does it happen? That unlike what we thought, the inhibitor does not block the active site of the enzyme, but rather it remodels the enzyme by creating a tunnel in which additional binding sites for the oligosaccharides are created. So it means that there is a longer substrate binding site. This kind of mechanism increases the affinity of the complex for the longer oligogalactronides, favoring their formation. So we have a system in which the plant has evolved a system to, this is what we already knew, subvert a pathogenesis factor into an elicitor defense responses, and this explained the change of substrate specificity of the product profile of the complex. Here is, there was an editorial on Science about this. Here is the complex that really can favor the interaction with longer fragments, favoring the production of the elicitor active oligogalactronides. And the novelty of the system is this a mixed organ enzyme in plant defense, so a new biological mechanism involved in the production of DAMPs. So we have a system that favors the production of DAMPs. And there is another intriguing paper in 2024, just again in January. And I show you this because this means that the work and the notion of DAMPs is sort of exploding in the plant field. What it was found, well, RALFs are peptides that regulate developmental responses, also inhibit immunity. What it was found that complexes between these peptides and the oligogalactronides are formed, and these undergo phase separation, leading to a massive endocytosis of several receptors. It's a new process. And all this is mediated by specific receptor kinase named Feronia. This is a paper on Cells, January 2024. And here is the illustration of how the system works. Here we have the pectin, the homogalactronan, the breakage leaves the formation to the oligogalactronides. These are the RALF. There is phase separation. And there is this massive endocytosis induced by DAMPs and this RALF. So we don't understand really well now the significance, but this means really that oligogalactronides are quite relevant in many aspects, a unique biological design for their formation, unusual, unexpected roles in signaling that have to be clarified, elucidated. So really, they are emerging as important, but we have always thought that they were important. You know, sometimes DAMPs are really overlooked. And I had a lot of colleagues saying, I don't believe in OGs several times. I don't think that they can play a role. They are signals. They are just anionic fragments. Well, we were not convinced of that. And years ago, we designed a protein fusion between a polygalactronase and a PGIP that we conceived as an OG-forming machine. I can say that the name was so appropriate in the light of what it was found from the structural work. And so we called it an OG machine. And we studied the plants expressing in an inducible way the OG machine are more resistant to pathogen, and they have activated defense responses because they have higher levels of OG in the tissue. We have all the expertise to measure those by chromatography and mass spectrometry. The point is that when expressed at high levels, OG cause deleterious immunity. The stop growth, plants can even die. So, leading to the idea that OG signaling by oligogalactronides, being endogenous signals, must be strictly controlled not to have deleterious effect. So here we have this problem with the endogenous signals, with the DAMPs, that can be dangerous. They come from a normal component of the cell, but they can be dangerous. And their formation should be strictly controlled, otherwise it goes out of control easily. And so we were thinking about homeostatic control of their activity, either during immunity to return from defense to growth. This is the well-known growth-defense trade-off, but also during growth, because the cell wall from which the oligogalacturide derive is remodeled during development. And in that case, you also have production of oligogalacturonides. So how can this be accomplished, this homeostatic control? The OG machine plants, the plants expressing the OG machine, were instrumental. And because there we found the presence of fragments that were similar to OG, but they were not OGs. And by mass spectrometry, we found out that they were oxidized OGs. This discovery led us to the further discovery that there are enzymes in plants that oxidize the reducing end of damp-active oligosaccharides. And by doing so, they also produce H2O2. So here we have ROS coming out. And this was true for oligogalacturonides and for cellulose fragments. And also for hemicellulose fragments. So we discovered this enzyme, at least for oligogalacturonides oxidases, and at least to cellulose fragments oxidases. So we have been working on it lately. And just to make a long story short, the oxidation sort of nearly inactivates the DAMPs, and this has been published. And overexpression and disruption of the oligogalacturonide oxidases in plants by transgenic means has an impact on response to pathogen and response to wounding as well, so to tissue injury. So they really behave in the direction we think as elements, these enzymes, as elements that maintain homeostasis, that bring back an exaggerated response. And just in line with this statement, when the party is over, cleanup begins. And so really the return to the homeostasis is very important. We have also evidence that their alteration also affects developmental processes. This is the conclusion. So this enzyme, the berberine bridge enzyme-like are oxidases that control the homeostasis of different classes of cell wall-derived DAMPs, keeping the immunity under control, also regulating development. And this indicates that they work at the crossroad between defense and development. Last slide, but the comments may be more. Of course, there are a lot of questions, a lot to do, how all these enzymes and DAMPs work in orchestrating immunity. The system might be very complex. What is their role in this ancestral immune response that probably is at the origin of the immune system? How do they work in the wound response and wound healing? And up here, no? What am I doing? Yes. And another key aspect is how do they work in grafting? Because grafting is a very common practice in plants. And at the beginning of the healing process, at the junction, there is a large production of homogalacturonan, the source of OGs. And now we are investigating how these DAMPs participate in the grafting process and the healing process. Of course, we are studying also the role in development. And in all this, what is the meaning of this ROS production, H2O2? We have a big production by oligogalacturonides, but this enzyme can produce ROS in a very localized way. So when you turn off, then you have this localized production of ROS can be important. So, and how the system evolve, and this is another key question. Just a really acknowledgement to the group. But I want just to finish saying something about a concept that already emerged. A recent paper showed that the maximal immune response occurs when there is cell wall damage, when there is tissue damage. So tissue injury and the activation of immune response go together. The immune response is very low if there is no, for example, remodeling of the cell wall. And this calls a role of these cell wall-derived enzymes. So the maximal defense response comes when you have cell wall remodeling and then potential release of DAMPs. And also the response to MAMPs just follows the same path. If you don't have damage, and you have only the bacteria, very little immune response. You need to have both. And so with this, I just stop, and there are many things to say, but I know I am an ectopic lecturer here. So anyway, this is what I wanted to tell you.
Yeah, biological objects that are so far away from us react in just a similar way. There are certainly questions from the audience.
Yeah, the innate immune system is thought to be evolved 1 billion years ago. And so that's why we find it so spread with similar strategies.
Questions from the audience? Polly.
I first heard in Mexico from you about the remodeling of the fungal virulence factor into an initiator of immunity. Right? And I've been thinking ever since, is there anything like that in animals? I have not found anything. Have you?
Not yet. And also because I told you, there is sort of distance from the concept of DAMs, of the importance of DAMs, of the importance of oligogalactonides or fragments. So let's say that no, that's why maybe we can learn something, maybe there is something, and something similar, yes. And the notion that is a specific biological design came just now. So it means that really in the evolution, there has been really the evolution of a specific structural mechanism to favor the DAMs. And this, of course, points to a very important role. And the roles are really emerging, so we are very excited about that, but still there is a war to be discovered because the system is very complex. And the other things that we don't know if we exist is whether oxidation is also a relevant mechanism in animal immunity. Oxidation, so inactivation by oxidation. We try to do something with hyaluronan fragments, but very, very sporadically, so actually the experiments were not really well performed.
Any questions from the audience? Yeah, please.
Completely stupid question. Do plants have an coagulation system?
Sorry?
Do plants have a coagulation system?
Coagulation.
Well, not coagulation, but they have healing processes. And actually, in wounding or in transplant, it's a joining. In wounding, you have the healing of the tissue. It's not coagulation, there is nothing similar, but there is a process of which very little is yet known that heals all the surface because we published a paper. A wound is a wonderful passage for pathogens, so the tissue must be sealed, otherwise it's too dangerous. And that's why that the response to wounding is really evolutionary, more ancient. So the plants must have learned first to defend themselves from tissue injury, and then combine this with the danger represented by microbes.
More questions from the audience? If this is not the case, I thank you very much. I think it was a highly interesting talk, especially for us being so concentrated on a certain topic in humans called transplantation. And now we are led far away from that system and see that there are similar processes going on in plants. Thank you.
Let's look at that. When you graft, can you graft any plant to any? No.
Incompatibility.
So,
and the basis of the incompatibility is one, the elucidation of the incompatibility is another challenge. And now we have some tools to investigate whether these dams can play a role in the incompatibility or in the compatibility. But that is genetically controlled. There are also receptors that have been identified, but the mechanistic processes are not really known.
So again, thank you, Giulia. And I want to not to keep you more away from our dinner. We are now at the end of this symposium, and I can say if you are celebrating a birthday party, usually there's presents, and I think we've had some very nice and surprising presents for tonight. And we've had two very interesting speakers that made us, how do you say, über den Tellerrand schauen, look out of the box. And this is really something that we can say was a surprise present. So I thank you for the presents, and I invite you all now for a nice dinner. We are going downstairs and are having a 3-course meal, and everybody can decide what kind of main course he or she wants to have. So this is the end of the meeting. Thank you.